Hello, my name is Amanda Febriyanti Nurdevi. I'm an undergraduate student at IPB University majoring in plant protection. So I'd like to talk about CRISPR or Cas9 mediated genome editing in garlic against Onion Yellow Dwarf Virus. Ladies and gentlemen, have you ever typed an email to your professor and sometimes you got some typo in it? Then your professor might misunderstand the messages that you're trying to tell him. So what you're gonna do is to press the big space button on the keyboard, correcting and editing the typo before the email being sent to your professor. So what I want to say is this editing can also be done at the molecular level, where we took genome sequences, then we analyze to correct the typo like we used to do in our keyboard, so that we can manipulate genes to give a specific trait in our organisms. One of the discoveries in genetic engineering is CRISPR technology, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. This technology is used in plant protection as a method to produce plants that are resistant to pests, disease, and abiotic stress. CRISPR method has already been applied to create virus-resistant plants. For example, potato that has resistance to potaviruses such as potavirus Y that have been successfully developed using the CRISPR method. To date, CRISPR-mediated genome editing in garlic crop has not been reported. Yet, garlic is an important crop due to its various benefits. However, local garlic production is decreasing due to plant health issues, especially plant viruses, which can cause the decreased garlic yields in Indonesia. In this study, I utilized CRISPR technology as gene therapy approach against important viruses in garlic, such as Onion Yellow Dwarf Virus or OYDV. This research begins with the identification of target genes that play a role in garlic resistance to OYDV, specifically genes that encode proteins necessary for the virus to replicate. There are two main components required to CRISPR method. There is a CRISPR-associated protein 9 or Cas9 enzymes, which acts as a molecular scissors, and a guide RNA which called gRNA or sgRNA that will search for nucleotide sequence of the harmful RNA that corresponds to the target sequence. Then it will direct Cas9 to the specific sequence that needs to be altered. If the guide RNA and Cas9 is introduced into the target cells, it will bind to the target sequence so the genome editing may occur at that location by modifying, deleting, or inserting new sequences. The altered genome will encode a non-functional protein that is not specific for OYDV to replicate in garlic cells. As a result, garlic becomes resistant to the virus, and by this method, we can produce resistant garlic to OYDV.